University. I'm going to talk to you today about a bioinformatic approach that we've been working on in collaboration with the Dresch lab, thinking about nucleotide interdependency when we're trying to predict these transcription factor binding sites. So first I want to start with just kind of the simple idea that's really been in my head for the last 5 or 10 years now. This idea that I think all of you have seen as biologists, right? Here's your central dogma. We have DNA being transcribed to RNA, being translated to protein. But really, what we're kind of thinking about, what's most important in my lab, is this idea that proteins then can kind of feed back and bind onto other segments of DNA to help regulate this transcriptional process. So really, what we've been focusing on is trying to figure out how these proteins can recognize places in the genome to bind. And we like to kind of think of this as the grammar of transcriptional regulation, right? Just like you would think of grammar of the English language, right? Where do you put your nouns, where do you put your verbs, where do you put your transcription factors? So thinking about this kind of just in terms of a simple genome, the fruit fly genome that we study, there's approximately 140 million base pairs that we're looking at. So A's, C's, G's, and T's. And we need to somehow look at this genome and predict where our favorite transcription factors bind. Right? Not a trivial question to answer. So thinking about it kind of this way, proteins bind typically in a sequence-specific manner, finding binding sites that are somewhere between 5 and 15 base pairs. And of course, you can talk to different people who may argue that binding sites are actually longer than 15 base pairs for certain proteins. Most of the transcription factors we're interested in are fairly well studied, and people do agree that the binding sites are somewhere between 5 and 15 base pairs. But still, these transcription factors need to find these binding sites among millions of base pairs in the genome. So some of the questions that we've been kind of thinking about and trying to answer are, first of all, how do we know where they bind? Second of all, what is the specificity, the sequence specificity, or kind of thinking of this more from a mathematical or physical perspective, what is the binding affinity? You may have many places in the genome where you can bind, but you might bind better or stronger, right, to some DNA sequences than others. And then, of course, the kind of buzzword you often hear in this field is consensus sequence. So I always like to think about the question, is there a single consensus site, or do transcription factors bind to many different sequences, and there is no real consensus, not one sequence they're looking for, but kind of a group of sequences that they may bind to just as well. So I'm going to start with kind of a traditional PWM approach. So for many years now, I mean this is probably about 30 or 40 years now, people have been using this kind of bioinformatic approach that's often referred to as a PWM, or a position weight matrix. And the big kind of underlying assumption here is that there's nucleotide independence. So what you would do is you would collect some data of known binding sites, and you would align those sequences. So this can be done using PBM, using footprint data. There are many approaches out there. But the idea being that you have some known binding sites for your protein of interest. You align them all, and then you can build this matrix where you say, okay, at position 1, what is the frequency I found in A, a C, a G, or a T? Then you move to position 2, right? And you now ask that same frequency question, completely ignoring the nucleotide you saw at position 1, right? This is where this term independence comes into play. So you're really thinking of each nucleotide in that sequence as being independent of all other nucleotides. So we can tally off these frequencies. We can calculate the probability of each base being at each position there. And often we illustrate this using these web logos And the way we've kind of thought about it in my group is, okay, so every nucleotide is important, but you may more often see an A in this position than anything else. So you see a big A in the squad logo. So traditional. Now, we started thinking about how we might change this kind of traditional approach to think about that assumption of independence. Are they really independent, right? Does it matter what that base is adjacent to me? Or maybe, right, what I really care about is a base 4 nucleotides away, right? So thinking about maybe, there is some interplay within a binding site. So here is kind of our model, or at least an example of our model. We start off very much in the same way. We have these known sequences, right, known binding sites. They're all aligned. But now we take a frame and we say, okay, well, for this example, we take a 6 base pair frame and we assume that the first nucleotide and the last nucleotide of that frame have some dependence on one another. So then we can build a similar matrix. But now notice this matrix, right, looks at all 6 mers, where the 4 nucleotides in between don't matter. We don't care what they are, we just care what those 2 outside nucleotides are, because we're assuming that they have some dependence on one another. And you build this model and think about shifting that 6 nucleotide frame all the way through your known binding sites, or your aligned sequences. Okay, now this example is one that always kind of makes me laugh that we put it up here, because if you look at your position weight matrix, you might notice right away, wait a minute, by using this approach, you're ignoring those 2 largest letters there, those 2 A's. And absolutely that is correct. This is just an example. So when we actually do this in practice, we're going to look at all possible combinations of dependence. So maybe those A's are just a coincidence, right? I doubt it though. So what we're going to start thinking about is not just 2 nucleotides depending on one another, but any number within that frame. So here is the algorithm. We call this algorithm the Mars algorithm. And the way we designed this was to do it in the most kind of unbiased way possible. So the way we design the models are we take all integer values between 0 and 31 and their binary representations. So what we've really done is we've just come up with every possible binary string of length 6. Then we rewrite this by replacing every 0 with a K and every 1 with an M. We then remove all of our leading Ks and add an M to the end. Now the reason for this is that we're thinking of M's as the nucleotides that matter, that give us some information about our binding site. Ks are going to be those nucleotides that we ignore, that are not giving us much information about our binding sites. So we remove leading Ks because you don't care about them. We can throw them away at the beginning. And we add an M to make sure that every single model has at least one nucleotide that contributes to the affinity of that binding site. So in kind of summary, we refer to these as gapped N-mers. The final column here gives you all possible gapped N-mers of length less than or equal to 6. Meaning, we have different combinations of nucleotide dependence for every possible string of length less than or equal to 6. So we're trying to do this kind of combinatorially in an unbiased way. Now, of course, you can't build an algorithm without testing it, right? So we needed some experimental data to test this on. Luckily, I had been collaborating with the Drool lab, and we had been doing a lot of work in Drosophila, and thinking about binding sites in Drosophila, and there's a wealth of data out there for the transcription factors involved in early development. So we started just by looking at these 20 transcription factors, and notice that they're not all transcription factors that are even regulating the same axis. So if you look at this third column here, the classification tells you that we took some transcription factors that are regulating the anterior-posterior axis, some that are regulating dorsal-ventral, and some that are considered parallel genes. So we really tried to take a kind of variety of transcription factors from early Drosophila development that we had kind of comparative or similar data for. And the goal in this whole project was to ask the question, well, okay, if I build these models using known binding sites from footprint data, then, right, I put those models aside and now take chip data, so known chip peaks for these particular transcription factors. Can our algorithm, can our model correctly identify those chip peaks as having binding sites in them, right? And when we do this, we always compare our chip peak data to scrambled data, or kind of random DNA sequences. So really the question is, can you correctly identify true chip peaks from false chip peaks, right? And we came up with a scoring system. I won't get into the whole derivation of that scoring system. I'll just give you kind of the bare bones here. An RZ score above 0.5 means that the algorithm is correctly identifying true chip peaks opposed to false ones. A score of less than 0.5 means it incorrectly identifies those false chip peaks as true chip peaks more often. And if an RZ score is exactly at 0.5, then the model is really not doing any better than a random guess. So here are the results. And what I'm showing you here is just for 15 transcription factors, so 15 of those. We see these results, and I will point out very quickly, the axes here, not to confuse you, the y-axis is that RZ score. So you'll notice there's a line across each one. That is that line of 0.5. So any data point above that line means that it's doing better than random, okay? The x-axis here corresponds to the fact that we ran this model using many different thresholds. So what is a binding site, right? I can score any DNA sequence, but I need to then take that score and threshold it to determine whether I actually predicted a binding site or not from my model. So the way you want to read this x-axis is on the left is the lowest threshold position. So that would be where I'm predicting all binding sites, whether they're weak binding sites or strong binding sites. As you move to the right, I'm only considering the strongest binding sites to be true binding sites. This was a question we wanted to ask. Do the models perform differently if you only consider strong binding sites, or if you were to throw in all of your possible binding sites into that training data? And from here, it might be obvious to your eye that some transcription factors, you seem to see a lot of blue on these graphs, and some transcription factors seem to kind of flatline or have just a very slight monotonic decrease. So we grouped these into two separate categories. We said, oh, the TS that we see fall into roughly two groups. The first group, what I think of as the kind of uninteresting group, is a group, and just to blow it up, this is what most of them look like. So what you're seeing here on these dots and whiskers is you're seeing all 32 models, and you're seeing the deviation between those 32 models. So you're not seeing much of a difference between using the model with that independent assumption versus models with many kind of complicated dependencies in there. You see the models do slightly better at lower thresholds when you include all binding sites. It's easier to find those chip peaks. And they don't do much better than random when you only consider the very strongest binding sites. Now the second group of transcription factors, so here's an example, purple. We are seeing quite a difference as you move across. So first of all, if I were to pick a single threshold position, I see a lot of blue there, right? I see a lot of variation between those different models, between those different assumptions. So what we think is probably going on here is there actually is quite a bit of interdependency between those nucleotides. that this independent assumption we've been using is maybe not the best assumption if you're trying to predict true binding sites. Also, you notice there's quite a bit of change, right, in those scores as we go from lower threshold to higher threshold again. So you do have to be careful whether we want to consider all possible binding sites, weak binding sites, strong binding sites, or whether we just want to consider those binding sites with the highest affinity. And depending on what you want to use this for, what application, you may say, I want every possible binding site, I want to be able to predict every chip leak out there. Or you may say, well, I only want to study CRMs, right, transcriptional regulation, when there's a very strong binding site driving that regulation. All right, so the two groups that they fell into, what I refer to as group A and group B, were these two groups here. Just looking at the kind of more interesting group again, that group B, where we do seem to see some interdependency. You can look at the top 5 performing models, and for some reason these labels didn't come out here. But just so I can kind of point this out to you, these are the best performing models along with the traditional approaches. And drawing it up here so that you can really see model by model, there are quite large variations here. So the performance really is different when you have nucleotide interdependency versus that independent assumption. Then we went one step farther and we looked at one of those transcription factors, Pruppel. Again, one of the ones that we've been looking at in the Hox gene complex. We have a pretty good idea that Pruppel is repressing, right? Acts as a repressor in IB5 and IB7B. In the past, in the late 90s, quite a few people had studied Pruppel as a repressor also in a very well-known CRM, Eve-Strike2. So taking these 3 CRMs, we went through and we said, well, how does our model do, or our different models, do on predicting Pruppel binding sites in these 3 CRMs where we have a very good idea from molecular dissection where those binding sites are. And these numbers were kind of amazing to me because what you're seeing here is you're seeing, okay, the traditional matrix, the PWM matrix M, it finds, in this case, all 3 true binding sites, but it finds 630 false positives. So it's basically just a very promiscuous model, right? It's predicting binding sites everywhere. While in Eve-Strike2, we find only 2 of those 3 Pruppel sites, but the number of false positives is at 17, dramatically lower. And over the Hox complex, these 2 enhancers, IB5 and IB7B, we actually found that having nucleotide dependence here was still able to find all true positives and reduce the number of false positives by orders of magnitude. So, in kind of conclusion here, right, this is really kind of leading me to believe that using these models with nucleotide dependency can really help narrow down where those CRM regions are and where those important binding sites that we may then want to go in and mutate to try and dissect out what's going on in that enhancer region. So, in conclusion, I'll say the degree of nucleotide interdependency seems to vary from TF to TF. Right? We found these 2 groups, and 10 out of 15 didn't show much interdependency, while those other 5 in group B did seem to show quite a big difference between model assumptions, and also the best performing models seem to show quite a bit of interdependency there. For Pruppel, we found that the more complex gapped enmer seemed to outperform the traditional PWM as well as a dinucleotide model on known enhancers where we have known binding sites. And the only question that this always kind of leaves me with is, how universal is this? Right? All it really talked about is 15 transcription factors in Drosophila development. Right? So often, especially coming from the math department, people will say, who cares about fruit flies? Why are you studying fruit flies? Why are you studying these 15 transcription factors? How universal are these rules really? How applicable? So I'm just going to leave you with this one figure. This is from a mammalian study where they looked at transcription factors in mice, and they looked at over 30 different models, and they found that what you're seeing is all of this purple shows that actually most of these models perform very similarly on the majority of transcription factors. Then, if you look over here to the right, you'll find there are a small subset of transcription factors that, looking at those different model assumptions, really changes their performance. So I think maybe we are onto something here. It may not be every transcription factor that needs these kind of more complicated models, but there are a subset of them that I think we could really improve predictions by using these different assumptions and thinking about kind of different types of models outside of the box. So I will stop. I know I'm on time. I would like to acknowledge, though, my collaborator, Robert Droll, again, and I had a student at Harvey Mudd when I was there a couple years ago, Rowan Zellers, and he was really kind of the machine behind this project. He was an amazing undergrad. He coded up everything, did almost all the computation, so I just wanted to make sure I mentioned him and some of my funding sources currently. I'd be happy to take any questions.
So maybe I ask a question. So this interdependency, maybe I missed it, but we usually work with 6 base pair recognition sites. Yeah.
Mm-hmm.
So what did you find? Which nucleotides are interdependent on each other? Is there a positional preference?
So that's a really good question. So I would say with the 5 transcription factors that we did find a lot of interdependency with, we found different gapped N-mers, different kind of 6-mers that were performing well on the 5 transcription factors, although what we did notice was there is this kind of signature pattern within each of them. We found an MKKM. So it seems like there is definitely something going on in that kind of 4 to 5 base pair gap. And we started trying to think a little bit about maybe binding domains. Is it something like a zinc finger? Is it maybe just the helix? Turn, thinking kind of, okay, well, if you turn in 10.5 base pairs, maybe half of a turn is where you're seeing this dependence. And really, I think to answer your question well, we would like to look at 100 or 200 more transcription factors and see if we can really kind of nail down what that might be.
Well, my question is related to a situation where you have multiple factors binding to identical sites, so you share. So do you have this assumption in your model that you can have more than one factor competing actually for the same site?
Oh yeah. So when we run these models, we can actually run this model for different transcription factors along the same DNA sequence. Absolutely. I will say the one problem we have there is how you relate some of these bioinformatic approaches back to true binding affinities, right? It's always a relative binding affinity. So the problem there is when we start thinking about competition between proteins, I can give you a binary answer. I can say 2 proteins want to bind to the same site, right? But it's very hard for me to say when 2 proteins want to bind to the same site, which one is binding with a stronger affinity.
Yeah, that's correct. So in terms of those variations that you see, that you cannot understand, that this may come from this type of variation that requires one level of variations added to the system.
Absolutely. So I don't know that I have a great answer to that. I think one thing that we've kind of thought about with my collaborator at Michigan State is if you can somehow break apart that binding site, right? If you know that two transcription factors want to bind to the same sequence, can you find a single mutation that will make one outcompete the other?
Yeah, that's correct actually. From experimental standpoint, it's correct.